Our next speaker is here in person. It's Christian Riedel from Karolinska Institute in Stockholm.
Thank you. That's it. All right. So I would like to start by thanking the organizers for the kind invitation to come here and present some of our latest research. And what I will tell you today about is a project that we started maybe roughly 4 years ago in my lab, where we've been mapping chromatin accessibility changes, in particular in long-lived insulin IGF signaling mutant animals. Yeah, I want to tell you about some outcomes from this work. How does one? Oh, that's it. Yeah. Okay. Good. So to begin, I just want to give you a bit of background on this project. And just remind you once again, even though you have already heard about this pathway, that insulin IGF signaling is really one of the most important and powerful aging regulatory pathways that you can find in metazones. It's a nutrient sensing pathway, and under normal growth conditions where nutrient levels are high, there will be insulin secreted in the organism, and so us, who we conduct our work in C. elegans, we then in target tissues have a receptor for insulin that is called DAF2 that then gets bound by this insulin and activated, and this then triggers a phosphoregulatory cascade that eventually leads to a phosphorylation of a downstream transcription factor that is called DAF16, or you also will know it as the ortholog to the FOXO family of transcription factors that you find in higher eukaryotes. Now, phosphorylation of DAF16 normally keeps it inactive because phosphorylation of DAF16 leads to sequestration by 14-3-3 proteins in the cytoplasm, and thus the transcription factor is away from its target genes. Now, under such normal, well-fed growth conditions, animals exhibit a normal, typical lifespan. In this experiment, it was about 15 days on average. However, the reason why this signaling pathway is so very important for aging regulation is that if you block it, and DAF16 is no longer getting phosphorylated, it can actually then enter the nucleus and there drive expression of target genes. And these are mostly stress resistance inducing and aging preventive genes that eventually make the organism much longer lived and healthier. And if we then look at the lifespan assay that we looked at before, you can see that the lifespan of the animals indeed increases 2- to 3-fold. Now, it's important to note, though, even though DAF16 is essential for DAF2 or for the longevity and reduced insulin IGF signaling mutants, by itself it's not sufficient to promote this phenotype or to promote this longevity. So, for example, if by mutating the phosphorylation sites on DAF16, you would prevent its sequestration by 14-3-3 proteins and create animals with constitutively nuclear DAF16, this is not sufficient to promote longevity. Or also, if you would overexpress this transcription factor, you don't get the same lifespan extension as if you were blocking insulin IGF signaling entirely. So one needs to conclude that other players and mechanisms are involved in conferring this longevity. And while many studies have already been published to date that describe such mechanisms, today I would like to focus with you on one set of mechanisms that relates to the chromatin landscape, because there we felt that they haven't been exhaustively explored yet. So here I would like to actually go back to a review from Lopez-Otin and colleagues from 2013 that described the hallmarks of aging, so phenotypes that are arising as we get older. And it's well established that the chromatin landscape changes as we get older. Now, the question we were interested in is now if the chromatin landscape not only changes as we get older, but if the chromatin landscape also changes in long-lived mutant conditions, for example, reduced insulin IGF signaling mutants, whether it's basically a hallmark of also those animals. If that's the case, the question then also arises if these chromatin changes are important for the animal's longevity. So in our research, we gained first evidence that chromatin changes are important for the longevity in reduced insulin IGF signaling mutant animals from a study where we looked for binding partners of this transcription factor that I mentioned before, of DAF16. So we were namely looking for factors that might help this transcription factor to turn on its target genes. And so what we did was to grow C. elegans to large scale. Then we immunoprecipitated the transcription factor from these animals and looked by mass spectrometry for proteins that would co-purify with DAF16, identified more than 130 of those, and then screened by RNAi through all these factors to look for factors that would not only bind to DAF16 but also be important for DAF16 to fulfill its physiological roles. In the end, we found about 32 such candidate proteins, and while not all of them were related to chromatin or chromatin changes, I want to just highlight one factor that we found there, and it's already a published story, and that was a chromatin remodeler called switch-SNF. So this is a protein complex that can utilize ATP to reposition or eject nucleosomes from DNA. So we found that when the transcription factor gets activated and enters the nucleus, it associates with this chromatin remodeler. And it's apparently an important event because if we're looking here at the gray lines, the normally present longevity in DAF2 mutant animals, which is the dotted line, it gets more or less entirely abolished if you knock down this chromatin remodeler. Ultimately, this was the model that we then arrived at as to what was happening. So this chromatin remodeler is so important for this DAF2 mutant longevity because DAF16 recruits it to its target promoters and there requires it to reposition or eject nucleosomes from these promoter regions. There opens up the chromatin and makes it more accessible for downstream transcriptional components like RNA pol2 that then has an easier time to come in and drive the expression of the target genes. Now, for the data I want to show you today, it was then clear that these changes are very important and they're chromatin related and they're occurring in DAF2 mutant animals, but we were wondering if there's any additional changes that occur in chromatin. And in order to get at that, we decided to eventually conduct a comprehensive analysis of the chromatin accessibility changes that occur in these long-lived DAF2 mutant animals. And for that, we used a technique called attack sequencing. This is a transposon-based method that basically identifies regions where the chromatin is particularly open and accessible. We combined the attack sequencing with also mRNA sequencing to understand wherever there is a chromatin accessibility change, if that also leads to gene expression changes in the vicinity to those areas. And then compiled all this data in an analysis pipeline that we newly developed. It's called Cactus, but it's still unpublished, that pipeline. I also should mention this is now work by 3 people from the lab, 2 postdocs, Luis Millán-Ariño and Jérôme Salignon, as well as Ellen Kavchek, a PhD student from the lab. So there's pretty much 2 types of events that you would be looking out for when conducting now such an attack sequencing experiment. When we're looking here at models of genome tracks, so to say here, there can be 2 types of events when we're comparing wild-type animals to DAF2 mutant animals. The one thing is shown on the left, that a region will initially be closed, but opens up in DAF2 mutant animals, or on the right, it can be a region that is open in wild-type animals, but then closes in DAF2 mutant animals. We call these regions also differentially accessible regions. What this then looks like in the genome browser, or the actual data, is shown here at the bottom. These are now 2 examples of regions where chromatin opened in DAF2 mutant animals. So you can see here, going from the light blue to the dark blue, how all of a sudden you get much more reads from these regions. When we now look genome-wide, what we eventually found was that there's actually a lot of regions where chromatin accessibility changes significantly in DAF2 mutant C. elegans. So there's about 3,000 regions where chromatin opens and 3,000 regions where chromatin closes. When we then look more closely as to which genome regions these changes occur in, the opening actually happens predominantly in enhancer regions, and to a lesser extent maybe in promoters. And the closing, if anything, occurs predominantly in 3' UTR regions. When we then try to correlate these events with gene expression changes, what we find is that, and we have here regions where chromatin opens at the top, or regions where chromatin closes at the bottom. The majority of places where chromatin opens or closes in DUF2 mutant animals, you cannot actually see any gene expression change in the vicinity. So maybe chromatin opens or closes, but this doesn't really change the ability of, for example, a transcription factor to come in or to get ejected. That would then affect changes in transcription in the vicinity. Nevertheless, there are, though, some, for the opening regions, some 632 regions where chromatin opens, and there is then gene induction in the vicinity, or some 486 regions where chromatin closes and there is a repression. So somehow here, potentially the binding of transcription factors or transcription machinery gets affected by these events. So we decided to focus on these regions and see if we can learn something from them. And the apparent hypothesis is that by these opening or closing events that lead to gene expression changes, what we could find there is maybe functionally important binding sites for transcription factors, and that these transcription factors could be contributors to the lifespan regulation in DUF2 mutant animals and could be contributing to their longevity. Looking at various angles of this, in the end, the most compelling information we got from looking at regions that were opening in DUF2 mutant animals. So these are, as you remember maybe from previous slides, predominantly enhancer and promoter regions. And when we look for transcription factor binding motifs that are enriched in these regions that are opening, we find this whole list of transcription factors whose motifs are enriched. It's encouraging to see that the motif for DUF16 is also there. But certainly we were interested in new transcription factors and then had to screen through the others. And we did that by RNAi, conducted lifespan assays to look for any of transcription factors that would specifically suppress the longevity of DUF2 mutants while not so much affecting lifespan of wild-type animals. And the candidate I want to focus on now with you is a Hox transcription factor called Lin39. You can maybe appreciate its abilities better here in such a longitudinal lifespan analysis. So when you knock it down, and this is the two blue lines in wild-type background animals, this doesn't have much of a lifespan phenotype or effect. However, if you do it in a DUF2 mutant background, this blocks the longevity or suppresses the longevity substantially. We then further found evidence that Lin39 really seems to have a lifespan regulatory capability by when we then also tried to overexpress this transcription factor, and overexpressing this transcription factor is actually sufficient in wild-type background to cause a modest lifespan extension of about 10%. So that's further encouraging that Lin39 must really be doing something lifespan regulatory. And the last data I would like to show you is then something also very interesting that we observed when we namely try to understand in which tissues this transcription factor is acting. You might think it acts maybe globally throughout the animal and it's important there to regulate lifespan. But what we actually find by tissue-specific RNAi, this is now initially here a whole animal RNAi experiment, and you see the suppression of the longevity. If we knock down Lin39 just in the muscle, there's maybe a moderate effect, but not much. If we knock it down in the hypodermis, there's not much happening. If we knock it down in the intestine, there's not much happening. But if we knock down Lin39 in neurons, we basically recapitulate the whole animal knockdown result, clearly underscoring that apparently Lin39 has a very tissue-specific role and influences lifespan by a function in neurons. So in summary, what I showed you now today is that chromatin changes are a hallmark not only of aging, but apparently also of long-lived animals with reduced insulin IGF signaling. I also showed you that chromatin accessibility changes in these long-lived animals seem to occur in particular in enhancer regions, which open. And then to a lesser extent, there's also changes occurring in promoter regions and 3' UTR regions. I introduced to you Lin39 as a transcription factor that seems to make use of this fact that certain chromatin regions are opening by binding to, in particular, enhancer regions that are opening up. And apparently this happens specifically in neurons, or at least that's where the important lifespan regulatory role arises. And so the interesting observation for us now comes from the literature when moving forward, where people imply that Lin39 might be important for neurons to maintain their identity with age. So our current working hypothesis is that the reason why Lin39 is important for DAF2 mutant animals to become long-lived is because they're expressed, or because it's acting in neurons that are required for the longevity of reduced insulin IGF signaling mutants. And if you have Lin39, this helps the cells to maintain their identity as the animal becomes old. However, if you remove Lin39, these neurons lose their identity and can no longer fulfill this function throughout life, and that's why the animals become shorter lived then. And so we're currently trying to validate this, and I'm also trying to pinpoint the neurons in which Lin39 might be acting. Okay, this brings me to the acknowledgements. I hope I didn't go over time too much. I want to thank the current lab members, several former lab members that contributed to the project, our collaborators, and our funding sources. And I thank you for your attention, and I'm happy to take questions.
Thank you very much, Christian. We have a question in the audience from Vera.
Hey.
Very exciting story. I'm curious towards the beginning of your talk where you were showing those Venn diagram, but there are lots of ataxic changes that didn't seem to lead anywhere. Mm-hmm. So I'm curious if there you analyzed only gene transcription, or did you look for things like transposons or other non-coding kind of element transcription?
No, for this, here we tried to initially keep it simple. We really just looked for expression changes in protein coding genes.
So because those may still be doing something.
They should be, but who knows? I mean, when you conduct chip-seq experiments, for example, that's also often an issue. Like, not everywhere where a transcription factor binds, it seems to be regulating the downstream genes. So, we might find at some of these places something interesting, but my prediction is not at all of them. Yeah.
Okay, we have also questions on Slack. One question which was upvoted is from Wilbert Vermey. He's asking why SNF interacts with the transcription factor 2H component P62, influencing nucleotide excision repair capacity, but also transcription. Can you elaborate on this with respect to your data?
I'm not sure I see a connection there instantly. I wouldn't have an answer to that, but I'm happy to chat with him more later on Slack.
Okay.
Maybe I need to think about this question some more.
Okay, so maybe we have time for one more question from Morten. So loss of SWI2 SNF2 in humans lead to premature aging. Are there multiple SWI-SNF in C. elegans, and are they equally important in aging longevity?
So this is a complex out of many subunits, the switch-NIF complex, and then you do have a number of ways in which this complex can be assembled. The catalytic core subunit in C. elegans is always the same, but there's accessory subunits that are interchangeable. We, in particular, in our study, found a phenotype for the buff type complex. I don't know if that tells you something, not the P buff, but that's about the depth to which we went.
Okay, thank you very much. Let's
Sure.
give Christian another round of applause. And